Anybody have any questions before we go? Questions about the term paper, outline, population genetics? Okay. So we're going to pick up where we left off last class, trying to both explain and then hopefully also predict what happens to allele frequencies over time given a few different variables, namely being the strength of selection and also dominance relationships. And that's some of what we started looking at last class. So again, a brief reminder, we started looking at plots like this that show how allele frequencies change over time, in this case giving different selection coefficients. So why is it that S of 0.3 has the fastest decline in frequency? You have an allele that's got a selection coefficient of 0.3. If it's currently at a frequency of 0.7 in a population, it drops the fastest. Why?
That's coefficient bigger.
So the coefficient is bigger, which means what? Pardon? More likely to disappear. So a big selection coefficient means reduced fitness. Right? So 30% reduction in fitness compared to the most fit genotype. So it seems to make sense that if you are worse off, if you've got a gene or an allele with a selection coefficient of 0.3, it's less fit. It will be eliminated by natural selection from the population the most rapidly. What would happen if there was a selection coefficient of 0? What would this graph look like then? So we've got one hypothesis that it might just be a horizontal line. It starts at 0.7, it stays at 0.7. It could increase. So what other factors change allele frequencies? Besides, so we're talking about natural selection here. That's one of the things that can cause allele frequency changes. So population size is critical. And that we discussed briefly last time is one thing that's not in these models that were used to prepare these plots. This is just based on selection, the act of natural selection changing allele frequencies. If we invoke anything else like mutation, small population size, then absolutely, and we'll see this in an upcoming chapter that's titled, unfortunately enough, Evolution in Real Populations, I think it's called. This is all artificial, theoretical work. So when we talk about evolution in real populations, then of course we have to let all of these things come into play, including population size. So you don't see smooth lines like this when you talk about real populations. You might see something where allele frequency goes up, it down, and then maybe it goes up to 1. Maybe it becomes fixed, 100% frequency. Maybe, maybe not. Maybe instead at this point it actually drifts down here and gets lost. Becomes 0%. So that's what we'll see when we talk about evolution in real populations. But absolutely, if it has a selection coefficient of 0, at this point we could just simply say we would expect the allele frequency not to change. There's no selection acting on that allele. Any questions or concerns about that point? So flat line is fine for now, when all you're considering is the effect of natural selection on allele frequency. If it's S is 0, it's not good, it's not bad, it's a neutral version of a gene, so you wouldn't expect the allele frequency to change. I wanted to clarify this point because I think I either misspoke or I was unclear about what it means to be frequency independent versus a frequency dependent form of selection. So in frequency independent selection, what that means is that the value of the magnitude of the benefit of the trait, the magnitude of the benefit of the fitness, doesn't have to do with how frequently you find that trait in the population. That is, if you have the beneficial trait, it doesn't matter if everybody else has the beneficial trait or if very few other individuals have the trait. The fitness is independent of how often you find that trait in a population. And in contrast, by the end of the day, we'll talk about the two forms of natural selection on the right, frequency dependent, where it actually matters. The value of S for the trait depends on whether or not it's a common trait or a rare trait. And so you can think of that in two ways. It's either how common a trait is or how rare it is. Is commonness a word? I'm going to make it a word. Frequency. So that's what we're going to finish talking about today. Overdominance, underdominance, and then positive and negative frequency-dependent selection. The last little point of reminder from last class, we wound up talking a lot about the difference between whether or not a trait was codominant or dominant and why it matters to selection when that really beneficial trait rises quickly, in frequency. So if there's a beneficial trait, it gets lost more rapidly when there's codominance. Because even in the heterozygote individual, some of the less useful trait is apparent. Because in codominance, you get what Lamarck might have called blending inheritance, and what others might have called blending inheritance, where the offspring looks like literally a combination of the phenotypes of the parents. And so the effect of that individual having the second allele, allele 2, which causes the lowest frequency or the lowest fitness, is somewhat apparent in the heterozygote. So the heterozygote can still be selected against because it has some of the negative trait. Whereas when allele 1, the new allele that's beneficial, is dominant, it masks the effect of that trait. Because if you have a heterozygote that harbors one of those less useful traits like allele 2, or the less useful alleles like allele 2, that individual has the same fitness as a homozygote for the good allele. So there's no way for natural selection to get rid of allele 2 from heterozygotes because they have just as much fitness as the homozygote allele 1 over allele 1. And that explains why when you have a codominant trait, it fixes much more quickly up here. So the trait goes to 100% frequency almost immediately, whereas it takes in dominance much longer for that trait to eventually, that second allele, allele 1, to eventually be eliminated from the population. So all dynamics related to dominance, codominance, recessiveness, and hopefully you'll be able to predict if I showed these sorts of graphs, or if I told you which form of dominance it was, you'd be able to draw a plot relating the different forms, the different frequencies of alleles and how they would change over time under dominance, recessiveness, and codominance. In other words, you'll see this again. Any questions or concerns about details on this now that you've had the weekend to reflect or not reflect? I feel your pain. Okay. So now we get into some territory that's a little bit, that wasn't strange enough, bless you. We're talking about different types of dominance now, overdominance and underdominance. Overdominance and underdominance are kind of difficult conceptually to think about, so I've put a couple of examples in that might help explain what these concepts represent. So this is what overdominance looks like. If you have a new allele and it starts at high frequency, it goes down. If you have a new allele and it's at low frequency, it goes up over generations and they reach some sort of an intermediate frequency where they're both at stable frequencies. So one allele, if at high frequency, decreases, if at low frequency, increases, and there's some sort of threshold. And this is what you expect when the heterozygotes have a higher fitness than either the homozygote for allele 1 or the homozygote for allele 2, which seems sort of weird. So when is it beneficial to be a heterozygote? So this is often called, as it says up there underneath the title, heterozygote advantage, or this is also called in evolution balancing selection. That is, there's selection for both alleles at once, and so those reach, again, this equilibrium, that balance, that the population as a whole has some of allele 1 and some of allele 2, and neither gets removed by natural selection, because there's a benefit to being a heterozygote. So natural selection never gets rid of one of the alleles. That would lead to fitness basically decreasing in the population, which is not really what natural selection does. So what sort of a situation in the real world could you think of that involves heterozygote advantage or balancing selection? Okay, so a great example is sickle cell anemia. Like the one that is in every middle school, high school, college, graduate school textbook, where it's beneficial to have, depending on where you live, it's beneficial to be a heterozygote. So in sickle cell anemia, what's the story? Why is allele 1 over allele 1 homozygote less fit?
The two dominant genes causes one to be susceptible to malaria, I think, and then two recessive alleles causes someone to develop sickle cell.
Right. So if you have A1 over A1, and this is just arbitrary for this story, but let's say allele 1, if you're homozygote, you're susceptible to getting malaria. If you have allele 2 over allele 2, then you get sickle cell anemia. So both of those traits indicate why the homozygotes are less fit. On one side, if you live in the certain area of the world where malaria is prevalent, you don't want to be homozygous for allele 1. But at the same time, you don't want to be homozygous for allele 2 because then you get sickle cell anemia, which presumably reduces your fitness as well. But heterozygotes get the benefit of allele 1 in that having just a single copy of allele 1 prevents susceptibility or prevents malaria. It makes you less susceptible to malaria. But the presence of the wild type allele 2 means that you don't get sickle cell anemia. This is one of many examples of heterozygote advantage. This is also known in a lot of plant species, hybrid vigor. When you have organisms that normally are inbreeding, that you would have the homozygotes by themselves are okay, like these. They have reasonable fitness. But when you cross two strains or two populations or two races of plants or two versions, two varieties of a plant, then all of a sudden their hybrids make even more fruit or bigger fruit or something like that. And that's the same idea, hybrid vigor. Then we have practically the opposite thing. Which is even more bizarre, I think. It's almost the exact opposite scenario. You can start with a population that has two alleles at intermediate frequencies. Like here, you have an allele that starts maybe at a frequency of 0.36 or something like that. If it's just above this arbitrary, it's not an arbitrary threshold. It's a threshold that will differ depending on the species and the allele you're looking at. There's some threshold. And if the allele frequency starts above that threshold, it will go to fixation. It'll go to 100% frequency. But if it starts below that threshold, which we would have to empirically determine, we'd have to collect data and find out where that imaginary threshold is. If the allele frequency is too small, it gets lost. And that's because it's bad to be a heterozygote. So in the graph on the left here, you'll see that it's the opposite pattern from overdominance, where it's better to be a homozygote than a heterozygote. So heterozygote disadvantage. get the lowest fitness if you happen to have two different alleles. Which is practically the opposite of everything we get told in science, that diversity is beneficial. But this is one case in which it's actually detrimental to have diversity, two different alleles brought together into the same individual. Does anybody know of any examples of this? So sickle cell is good for overdominance. When is it bad to be a heterozygote?
Maybe that allele's not needed. It might be too costly to have that tree.
So if it's so if one allele is costly, then would it be more costly to be homozygous for that allele? Would be my response. I'm not saying no, but it's something else we'd have to consider. So this is something we almost never talk about in genetics or in evolution. Well, not yet in your careers possibly. Outbreeding depression. This is the opposite of inbreeding depression, which we think about a lot more frequently. Like why it's bad to mate with your cousin. Right? You're not supposed to inbreed for many reasons. This is the opposite. This is outbreeding depression. This is what happens when you take two homozygous individuals that are totally fine by themselves, but when you combine those individuals, you cross them together, they make a less fit offspring. All right. So outbreeding depression, when it's bad to have more diversity. This is an example from my species that I work with, a publication from, wow, almost a decade ago now, 2008. And what these authors did was they took populations of this species, C. briggsii, the nematode, from different parts of the world, and they mated them together and they measured the fitness of the offspring. So every one of these groups of columns represents one cross. So here we have a strain called ED3083 and one called AF16 here on the left. And what they did to measure fitness was just calculate, in this case, how many of their offspring die before they hatch. So worms lay embryos that are basically in an eggshell, and they're asking, do the larvae hatch out of the eggs or not? So you'll see, naturally, in this one strain, ED3083, by itself, about 15% of their embryos don't hatch. So that's normal for this strain. Like almost a fifth of their kids, they produce an embryo, they don't hatch. They die before hatching. And in this other strain, AF16, it's like 2%. So which strain would you rather be? Which one's more fit? AF16. Like more of their offspring are going to probably survive to adulthood to reproduce themselves. And then what these authors did was they crossed ED3083 and AF16 together to make the hybrid. So just an F1. So half of their chromosomes came from, they're diploid, so one set of chromosomes came from ED3083, the other half from AF16. What happens? What's the fitness of the F1 hybrid?
Almost an average.
It's almost average. And so one of the things that the prediction, whenever you make a cross in terms of fitness, is that you expect to see, like, I will abbreviate here, MPA, mid-parent or average. So this looks like exactly what you'd expect. About half of the fitness of, compared to AF16, and twice the fitness almost of ED3083. So the hybrid kind of looks like an average of the parents. Where does that not occur on this chart? Where do we not see that sort of pattern where the dark gray bar is intermediate between the two parents? So yeah, right here, 3083 and 3034. What happens? Both of the parents have high embryonic lethality. But what happens when you make a hybrid? It drops. So what sort of selection does that look like?
Overdominance.
So it's better to be a hybrid. So that looks like overdominance, the example we just talked about a couple of slides ago. Okay, so now you should be asking, well, why is he showing us this slide if this is supposed to be talking about underdominance? So somebody find me an example of underdominance.
You have 16 in HK.
Yeah, the last one. If you cross the populations AF16 and HK104, they both have a very small frequency of of embryonic lethality, but when you mate them together, their F1 hybrids themselves lay a lot of eggs that significantly increased number of embryos, that asterisk, statistically significantly different, increase in embryonic lethality in their F1s. So that's an example of outbreeding depression. It's bad to be heterozygous, by definition. They cross two strains together to make someone who has more genetic diversity, some worm, and that worm is sick relative to the parents. They're less healthy than the parents. And that's what we study in my research lab. So this is why I get all excited about the AF16 HK104 cross. My students, both masters and undergraduate, work with those strains almost exclusively, trying to figure out why is it, what are the genes, what are the genetic differences between HK104 and AF16 that make their kids less healthy than the parents. Some genetic combination of alleles passed from AF16 and HK104 combine in an F1 make their kids less healthy. So that's underdominance. And by the way, I'll put a plug in for this. If you're not sure, and I think I mentioned this before, but if you don't know what you're going to do next year and you're thinking about a master's program, the application deadline for the biology master's program here has been extended. So it's currently open and will be open for a few more weeks. And if you're interested in doing some evolutionary genetics research, come talk to me. I've got spots. So that takes care of frequency-independent selection. Allele frequencies change, and they change in different patterns. They either go to fixation, or they go to an intermediate phenotype, depending on whether or not you're talking about directional selection. That's the one where the alleles either go to fixation, 100%, or loss, 0%, or if they reach an intermediate frequency. Then we have to talk about positive and negative frequency-dependent selection, which are really bizarre sorts of cases. Hopefully you read about these in your textbook. I'm going to use one example from the textbook and one example that you've never heard of before probably to illustrate these 2 points. Why would the magnitude of the benefit or detriment of having an allele depend on how common that allele is? So positive frequency-dependent selection means if the frequency of the trait, if the number of individuals that have that trait is high, then everybody benefits from having that trait. There's a positive relationship. More people have the trait, more people have individuals have higher fitness.
Maybe something to do with mating would be a good one.
So maybe something to do with mating.
So if everybody has a trait, being able to look for that trait specifically.
Okay, so if all the males say have a particular trait the females select for, then everybody benefits. So when is it good to have the common trait? When is it bad to be an outlier? When is it bad to be rare? So these are two different graphs that you can look at in the textbook if you like. I won't go over them in exquisite detail, but they show two different things. The one on the left is a graph of fitness. So this indicates exactly what we're talking about. That is, the frequency of the first allele increases, goes up to 100%. so does the fitness increase. So fitness and frequency of the allele have a positive relationship, a positive slope. So that's what we're talking about here. And the example that your textbook uses, and again, these are kind of rare. They might be very common in nature, but it's rare to find a good example to try to explain why positive frequency-dependent selection might actually exist. But absolutely, mating's a great example. So as the trait frequency increases, your fitness increases. Snails have an issue with having sex. Their shells get in the way. Bummer for them. Yeah, I know. We've probably got similar issues too, but man, and the snails around my house are going crazy this time of year. I should probably go take my own pictures so I can stop using a textbook version. So what does positive frequency-dependent selection have to do with snails? What is it? What trait are we talking about when we talk about snails and mating, where it's good to have the common trait?
It has to do with the direction that the shells.
Indeed. So it has to do with the direction that the shells spiral, or the chirality. If you took organic chemistry and you don't like that term, forget I said chirality. It has nothing to do with that. Well, it sort of does. It has to do with direction. So in snails, you want to mate with a partner that has the same direction of shell coiling that you do. Because that's the only way that the two, so these are both members with the same chirality, both individuals with the same chirality, so that they can mate together. If you happen to take a clockwise snail and a counterclockwise snail, they can't actually physically get in a position to mate because the shells prevent them from doing so. So as the frequency of being a counterclockwise shell, snail, increases in the population, more and more individuals in the population can freely mate with each other. So it's probably worst to have a population where the allele frequencies that cause different spiraling, counterclockwise and clockwise, when those two alleles are at the same frequency, 0.5 say, where half of the individuals have a shell that spirals in one direction and the other half spiral the other, you might imagine that that population is the least fit, because half of the individuals can't mate with the other half of the individuals in the population. So occasionally it's good to have the common trait, and it often, as was suggested earlier, has to do with mating. Yeah?
Does it have to do with the species as well? Different chirality shells are different species?
Well, that's an outstanding question. Do different species have different chirality of shells? Does a change in chirality of shells have anything to do with speciation? So we haven't talked about how species form yet, but this is a great example of how a species could form. If all of a sudden, in a population of entirely right-handed snails, there was a mutation, which is why this example is used, because there are alleles that cause one individual's chirality to reverse. So a single mutation can do this in these snails. So there is an allele that controls this trait. Yeah, if you got that mutation, if you were an individual that couldn't mate with clockwise coiling snails, for example, then you'd have to find somebody of your own chirality to mate with. And that would present a reproductive barrier between the counterclockwise snails and the clockwise snails. And you might predict at that point that never again in the future would the counterclockwise snails be able to mate with the clockwise or vice versa. And that would basically produce, over time, two different species, a counterclockwise species and a clockwise species. And we'll talk a lot more about the dynamics involved in species and how species form in a couple of weeks. But absolutely right. Anything that prevents individuals from mating with each other can be a cause of speciation. Other questions or concerns, comments? Everybody wants to go reclaim their extra hour of sleep. Man. So that's positive frequency dependent selection. If I was going to summarize that in one phrase or one sentence, I would say when it's good to be common. alleles that are beneficial when they're common. The opposite is an even more difficult story to tell. Or maybe not. Maybe you can help me out. When is it beneficial to have the really rare or unique phenotype? This is when allele frequencies are really low, that the owners of those rare alleles get the biggest fitness boost. Sexual selection. So
We could look different.
when you look different,
And that's what attracts them.
and that's what attracts a mate. Okay, so I think we're onto something. And the example that your textbook uses, I think, when I read this, I'll be perfectly honest, doesn't matter if you read it or not, I'll tell you what they said. But basically, the first time I read this, I thought, are you serious? This is a thing? They really went out on a limb to find an example of negative frequency dependent selection. So there's this species of fish where as it develops, its mouth is angled, so its mouth doesn't face straight ahead. It's either off to one side or the other. And this species of fish attacks other fish. They're scale-eating fish. So these fish attack other fish. But the fish always attack from one direction based on which side of their body their mouth is on. So if my mouth is on my right side of the body, I'd always attack other individuals on your left side, right? Because my mouth's on that side. So after a while, all of the prey species in the lake, the fish that are being attacked by this species, start to wise up. They're like, oh, they always attack from the left side because their mouths are always on the right. So they get used to watching to see if someone's going to attack them from the left side. So when is it beneficial to have the rare trait? So new mutation, you get this fish on the bottom where its mouth now develops to be slightly off center on the left. So it now attacks prey on their right sides, which they're not paying attention to because they're used to being attacked from the other direction. So this was the example that your textbook discusses of a case of negative frequency dependent selection, when it's good to have the rare or uncommon trait. Man. So is this common at all? I can't say because there aren't that many examples I know of negative frequency dependent selection. But I did find a second one. It also has to do with fish, but I'm not saying this necessarily means that only fish have negative frequency dependent selection. It also has to do with mating, much like positive frequency dependent selection did. So when is it good to have that rare trait when you're mating? These authors of this study didn't come to a conclusion per se, but they do tell a potentially applicable story at the end. So these are the killifish that they work with. And these are all males. And the reason that they were interested in studying this population of this species of fish is because these males have such varying color patterns, which you can see up here. There are tons of different mating colorations. So you've got males that have clear tails. You've got males with pigment on the lower half of the tail, the upper half of the tail, spots on their tail, tails that are mostly patterned or pigmented, different colors, huge variety. And so, the biologists started thinking, well, opposite to positive frequency dependent selection, where you might expect it's good to have the common trait because if females prefer one specific tail color or pattern, you'd want to have that pattern that the females are expecting to see. That's how you're going to get reproductive fitness. So how could you explain the maintenance of so much variation in male-specific coloration? Why would there be so many colors and patterns? How would females even know which one was the male that they wanted to mate with? What are the females selecting for you? And that's why they came up with this idea, well, maybe this is caused by negative frequency dependent selection. Maybe what females prefer here, ladies, tell me if this is true in humans, is somebody that's unique. Somebody that doesn't look like everybody else. So do we have examples of that in humans? When it's beneficial, when you're, guys or gals, ladies, gentlemen, whatever. are there cases where it's better for, you're more attracted to somebody who stands out than somebody who blends in? You want the guy with the flashy watch, the ginormous face, which is not what I've got. You like the guy with the mohawk, with the blue hair. Guys, you look for something unique or something that's common?
Uniqueness.
Pardon?
Uniqueness.
Okay, so let's go with uniqueness and see what happens. So in this population of fish, let's assume that fish are like humans. Most of the males, about 75% of the males, have an uncolored tail. And they've also got, so they classify the fish, the male fish, into are they uncolored or are they colored? Do the fins have mostly transparent area or do they mostly have color or pattern of any type? That doesn't matter what color or what pattern. And so they mixed artificially these males in different ratios. So they'd have a population, say an aquarium. They'd take 10 males. They'd put a large number of uncolored tailed males in and put one rare colored male in that aquarium. And they'd add some females and they'd find out who do the females choose to mate with, the common version or the rare version of the male. So what else do they have to do? That's one aquarium. They put a lot of clear-tailed, the transparent-tailed fish in, and they use a colored tail male as the rare male.
Switch up the ratios.
So they have to switch the ratios as well. So you have to set up a second aquarium where the common male is the colored tail male, and they put one transparent-tailed male in there as the rare male. So they did this actually in nature, in Trinidad, where these guppies live. They set up these pools in the actual rivers, and they just made sure they'd use nets to barricade any other males or females from going in or coming out. And so they put, instead of 10-to-1, as I described, they just used 3-to-1 ratios. Have a common male phenotype, and they'd have a rare male phenotype, and then afterwards, they took all of the F1 offspring that were produced by crosses between the females and the males, and they genotyped them. So they extracted the DNA from all of the parents, all of the males they put in, plus all of the females, and then they extracted DNA from all of their kids, and they compared the genotypes to figure out who was the dad. They didn't actually sit there and watch the matings happen. They just came back after the F1 individuals, the kids, were big enough to get DNA from. And then they did paternity testing, basically. And all they wanted to know was, who was the dad of more of the kids? Was it the rare male, or was it the males that had the more common phenotype? So here, the white areas are common. So here, we've got offspring. There was a small number of offspring sired by the common males, which there were more of, right? 3-to-1 ratio. There were 3 times as many common males mixed in with these females. Yet the vast majority of the offspring were sired by the rare male. And it didn't matter whether the rare male had the clear tail or the colored tail. They just had to be rare.
Dr. Ross?
Yeah.
I'm a little confused. What do you mean by the white common male?
Sorry, so the white bar up here indicates the common male was the father of that many offspring.
So that has nothing to do whether it had a clear tail or?
Correct, yes. So this was in both directions. So regardless of whether or not the rare male had the clear tail or the colored tail, it was just the white bars, columns rather, I guess, are representing the common phenotype males and how many kids they sired. And the black bars represent how many kids, whatever the rare phenotype was, how many kids they had. So almost twice as many, basically. And remember, there were way fewer of those rare males, too. So the authors conclude, at least in this species, females prefer to choose rare males, just because they're rare. There might be something else going on as well, but that was the conclusion of the study.
So the behavior of a female could be genetic? Or is it complete, well, it seems like it would be environmental, doesn't it?
So is it environmental? Is it genetic? What do you think?
But it's complex too, any behavior is complex.
So all of these experiments were done in the same environment, in nature, in these rivers. So they conclude it's genetic. But what is it that the females are actually, what is it? Are the females actually, saying, oh, you look different, you must be a good father? I don't know. I wish I knew how fish thought. Actually, I'm not sure that's true. Maybe I don't wish I knew fish think. So at the end of this story, the authors concluded a few things, possible reasons why negative frequency dependent selection, bad acronym, sorry, NFDS, why that might actually occur in this case. There are, so I didn't make up neophilia, it's a thing, you can Google it, check out the Wikipedia entry, it might be interesting. Attraction to new things. You have never seen this thing before. Flashy male. Hey, you're new, you're not from around here. Then these lower two actually have a longer history in science in terms of the rationale for why inbreeding avoidance and remating might actually be causes of negative frequency dependent selection. So one thing about being the rare individual in the population is that in some species, females do avoid mating with the same individual over and over again. So there's some reason to suspect that in females or males, depending on the species, you might have a trait evolved that causes you to prefer things that are rare. The second point in particular, inbreeding avoidance. So how do you tell whether or not you're mating with a cousin or a second cousin or a third cousin? If you're in a population that's highly localized, what would you do to tell who you're related to and who you're not related to?
Phenotypic?
Yeah, you'd look for new phenotypes. You're like, hey, everybody in this town looks the same. Hey, you look different. You must not be from around here. I'm going to mate with you.
Do they excrete the same chemicals?
So a lot of this may have to do with hormone signaling or some sort of sensory signaling that goes on aside from visual. So it could be partly visual, but it could be something else. There have been studies in probably many species. I only know of studies in stickleback fish where individuals, this has to do with immune systems now. So how do fish tell who would be a good parent? Oftentimes, depending on the species, individuals can assess how closely related they are to somebody else by picking up signals in these fish in the water that come from the immune system. So you can tell, do you have the same sorts of antibodies that I have? And somehow, female sticklebacks can tell which males they're presented with have more similar immune systems, and they prefer not to mate with them. So female three-spine stickleback fish can smell, taste, whatever. Chemical cues come from males that help them not mate with males that have the most similar immune systems. And presumably that's a signal of the benefit or the quality of the male. That if you have a kid that's a hybrid of you and your mate and their genomes, you might want to have a kid that's got a diverse immune system. I know that diversity comes after development, but there's also a genetic basis to diversity in the immune system. So that might be another sort of negative frequency dependent selection. So I'd like to leave you, well, I am going to leave you with a quick Socratic written exercise. This will be open for a couple hours. Don't write too much. But basically what I'd like you to do is to think about, everybody or almost everybody has picked a topic for the term paper. Take the last couple of minutes of class, reflect on whether or not any of this frequency dependent or frequency independent selection has to do with the trait that you picked. So just write like a couple sentences, maybe up to a paragraph. Just brainstorm. Doesn't even have to be full sentences. Did the trait you pick have something to do with what we talked about today? This will give you at least one thing, maybe, to write about in your paper, thinking about connections between material in class and your topic.